Restriction enzyme. Restriction enzyme, an enzyme from bacteria that can recognize specific base sequences in DNA and cut the DNA at that site, the restriction site. A restriction enzyme acts as a biochemical scissors, also called a restriction endonuclease. A restriction enzyme is a protein produced by bacteria that cleaves DNA at specific sites. Bacteria use restriction enzymes to defend against bacterial viruses called bacteriophages, or phage. When a phage infects a bacteria, it inserts its DNA into the bacteria so that it might be replicated. The restriction enzyme prevents replication of the phage DNA by cutting it into many pieces. Restriction enzymes were named for their ability to restrict or limit the number of strains of bacteriophage that can infect bacteria. Restriction enzymes can be isolated from bacteria and used in the laboratory to cut DNA. They are indispensable tools in recombinant DNA technology and genetic engineering. Each restriction enzyme recognizes a short specific sequence of nucleotide bases, the four basic chemical subunits of the linear double-stranded DNA molecule: adenine, cytosine, thymine, and guanine. These stretches in the DNA are called recognition sequences and are randomly distributed throughout the DNA. Different bacterial species make restriction enzymes that recognize different nucleotide sequences. After a restriction endonuclease recognizes a sequence, it cuts through the DNA molecule by catalyzing the hydrolysis, splitting of a chemical bond by addition of a water molecule, of the bond between adjacent nucleotides. Bacteria prevent their own DNA from being degraded in this manner by disguising their recognition sequences. Enzymes called methylases add methyl groups, CH3, to adenine or cytosine bases within the recognition sequence, which is thus modified and protected from the endonuclease. The restriction enzyme and its corresponding methylase constitute the restriction modification system of a bacterial species. All restriction enzymes are different. There are three classes of restriction enzymes, designated types I, second, and third. Types I and III enzymes are similar, in that both restriction and methylase activities are carried out by one large enzyme complex, in contrast to the type II system, in which the restriction enzyme is independent of its methylase. Type II restriction enzymes also differ from the other two types, in that they cleave DNA at specific sites within the recognition site. The others cleave DNA randomly, sometimes hundreds of bases from the recognition sequence. R E S T R I C T I O N E N Z Y M E. Restriction enzyme.